So again, like the focus of my work in the last couple of years has been in DNA language models with an emphasis on variant effect prediction. So what are DNA language models? So right now, most of you will be familiar with large language models such as GPT, powering ChatGPT. But what does it have to do with DNA? So the idea is that DNA is also basically, you can consider it as a language with four possible letters, A, C, G, T. And the idea of language modeling is to hide certain of these certain positions, certain nucleotides, and predict them from the context. Either in mass language model, they are basically, you can predict them from the context from the left and the right, whereas in causal language modeling, you only predict from left to right. Now, what is the task that you are trying to do? So based on a neural network, that can be, for example, a transformer, or more traditional in genomics, a convolutional neural network. This integrates all the information from around the mask position. And then a final layer could be like a linear projection that will predict the hidden nucleotide. So what can you do with this? So one thing is that you can evaluate the likelihood, let's say, given a new sequence with a variant, you can compute the likelihood. And usually I will dig into this in the next slides. But usually like a variant with a low likelihood will indicate that it might cause a disease. So it's finding that it is basically kind of an outlier. Now, you can also sample from the predicted distribution and generate new sequences. And also, you can basically use the representation of the sequence before the actual probabilities. So this kind of called embedding or latent representation, it can be adapted for different tasks, like using like additional data. If you have like gene annotation data or gene expression data, you can do fine-tuning, for example, and leverage all the patterns that have already been learned by the network in the language modeling task. So I will focus again on using the variant likelihood for variant effect prediction, in particular, predicting functional constraints. So now like a very brief description of variant effect prediction, what I mean by it. So there's millions of mutations in our genomes, and they contribute to like so many traits, from height to disease risk. But also, you know, in other really important systems such as our crops, they are really important for how these have been evolving and improved. So now a really like amazing feat of like of our technology is the ability to identify mutations, basically call variants in a genome. But still, we're very far from understanding which of them are impactful, which is usually like a few mutations in the millions are the ones that really have an impact. And now I wanted to also to mention that the focus of this work is on predicting, let's say, constraints, or it can also be called like fitness or deleteriousness, rather than predicting, for example, if it affects gene expression, which is something that requires additional data. So this is all unsupervised and just predicting constraints, similar to how conservation is used. So when basically the first approach that we tried using DNA language models, it can be described as follows. So this genome browser track displays the predicted probability by the model at different positions. For example, at the splice donor, if you, let's say, if you hide the G position here, the model already predicts that it's going to be a G there. And that is, as we know, the splice donor is super conserved. Now, if you look more in the, maybe more downstream in the intron, there are these positions that basically the model is supporting like a uniform distribution. It expresses that it has no preference. There is no evolutionary preference. So it would be like a neutral site. And similar, you see other, in general, for example, here, this codon, like the first two positions that are usually the ones that would change the amino acid, are predicted with more confidence than the third position that is usually just, maybe a mutation could just be synonymous and not change the amino acid. So the idea, something that we have shown is that it learns all these kind of motifs, not only in coding regions, but also in non-coding regions, such as transcription factor binding sites. And the idea is then to, again, to use the likelihoods of a variant to predict if it's going to be, if there's constraint or is it going to be a change in fitness of the sequence. For example, in the splice donor, let's say a mutation from the G to an A. So the A will have a very low probability. So that is what we're going to call as predicted as deleterious mutation. Whereas here in the intro on the right, maybe basically a mutation from C to A, that's going to have the same probability. So there's not going to be considered as functionally constrained. Now, another way to see this or to understand this is that the model is trained on healthy individuals, such as, for example, reference genomes. And when you finding mutations that cause disease, it could be basically similar to finding outliers. So sequences or specific mutations that don't seem to be from a healthy individual. So now an important caveat is that this is work that we did in the small genome of this plant, Arabidopsis, but it doesn't really work yet in the human genome. And you might think, well, maybe it's because the human genome is larger, it has more repeats, it has more long-range interactions, perhaps. There are many factors, and still, like maybe one, let's say, direct approach to try to solve this is to scale the model. Similar to how, let's say, there was GPT, then GPT-2, GPT-3, GPT-4. You always increase kind of the scale of the model and the data, and you get better models. But in genomics, it hasn't been that successful yet. So there's certainly been work from industry labs that use like hundreds of GPUs for a month. And it does improve a bit the performance, but it's still not that good, honestly, yet for this specific task of variant effect prediction. So we tried something similar rather than just scaling, kind of using more an inductive bias from biology. And specifically, what we looked into is multiple sequence alignments. So these are classic data structures in biology. So here what it represents is in this section from the genome, in this Mark4 gene, how the different species of vertebrates are aligned. And you can identify, again, you can see the GT, the splice donor motif is very conserved. And similar more in the intron, you can see there's substitutions, gaps, insertions. So it's really, if you can actually look at the evolutionary context of a sequence, that already gives you a lot of information that you can use. And one, note is that this has been first, has been championed in the protein modeling. If you think of AlphaFold, for example, it directly uses an MSA, a multiple sequence alignment, as input. And basically, it's kind of like bringing all the evolutionary context of a sequence into the model explicitly. And other models that also we inspired our work is MSA transformer. And these models have been found to be pretty efficient in the sense that even with a small model and even like a smaller amount of data, you can like beat larger models. So this is kind of our adaptation of these ideas to genomics. And the idea here is to take as inputs, again, the multiple sequence alignments, but just a window of the genome. We cannot model like 3 billion bases at a time. So it's actually really small, just 128 base pair window. And the idea is to, again, to mask a position in the genome, in the human genome, and predict it from the context, both the spatial context along the genome and the evolutionary context across different species. And we use a transformer neural network that will integrate all this information and output a probability distribution over the 4 nucleotides. And it is trained basically to maximize the probability of the reference. So to predict the reference, basically. There are some modifications that, such as some special weighting schemes and data augmentation that we do that we have in the supplements, and they are certainly interesting. But the main idea is trying to predict the reference genome. And now for variant effect prediction, to score a variant, we compute the log odds ratio between the 2 alleles, the alternate and the reference. And the intuition here is that for a neutral position, we expect the model to output a uniform distribution. So if it gives the same probability to alternate and reference, then you will have a log odds ratio of 0. That is what we will consider a neutral score of the model. But if the model gives a low probability to the alternate, such as finding that is really an outlier, then this score will be very negative. And that's what how we consider like a predicted to be like a functionally constrained site. So now an important question is, the evolutionary context is really powerful. Like there's so much information in just the specific alignment column. So does the model even use the spatial context? So to understand that, we did some experiments that I think show that it uses the spatial context as well. So one such experiment is looking at perfectly conserved positions. So these are basically simulated variants, because some of these are not observed in any living human, probably because they would be lethal. But still, we can just do simulated variants, and all of this, the model sees perfect alignment. However, we see that for synonymous variants, the scores are closer to 0. That is basically what we consider more benign. Whereas for missense, they are more negative or more deleterious. And again, for stop gain, that would, let's say, introduce truncate a protein, they are even more to the left, more negative or more deleterious. So the model is certainly, this is using the context to know if it's a synonymous or, let's say, a missense variant. This is not something that you can see from a single alignment column. And similarly, if we compare basically the scores, again, using this kind of log likelihood ratio, using either GPN-MSA or the column frequencies, they could be basically the empirical frequency in the MSA column to compute the log odds ratio, we see that for GPN-MSA, it has a more steep relationship with mean minor allele frequency, suggesting that basically a lower indicative score of GPN-MSA they tend to be like really constrained, so the allele frequency is lower. And basically lower also that what we will find by finding the lowest scores based just on the MSA column frequency. So now for benchmarking, we try to cover a wide range of different kinds of variants and different sources of evidence, such as clinical annotations or experimental data. So to begin with, for coding variants, we use ClinVar, that is a very established database of annotated pathogenic or disease-causing variants. And here, basically GPN-MSA does maybe a bit better than other methods, but we don't see much difference. So what this suggests is it is good for coding variants, business variant, but it's not providing that much value at this point compared to existing methods. That you could say that this task is somehow saturated in the sense that there are many models that are doing well at this task. Now, for moving on to regulatory variants, we found a set of curated disease-causing variants from OMIM, that is database of Mendelian disease variants. And here we see that GPN-MSA has a much larger gap with existing methods, such as CAD or PhyloP and FastCons. That's PhyloP and FastCons are conservation scores, whereas CAD combines annotations, a wide range of annotations, ranging from conservation to biochemical features and also population frequency data. So one drawback of OMIM is that it only has 400 known disease-causing variants. It's really small. So then we look at another database that is Nomad that has basically allele frequency. And we basically look to compare rare variants versus common variants, specifically singletons. And we know that this is not let's say, a hard label in the sense that singletons are not gonna be deleterious in general. The majority are probably not deleterious, but it will be enriched in deleterious mutations compared to common variants. So therefore, we use this odds ratio, a measure of enrichment, basically in the most extreme scores. Let's say you get like the lowest, let's say 1% or so, or more like 0.01% of scores. And we find that basically at this bin, it is 100 times more enriched in rare than common variants compared to the general distribution. And again, this is a pretty large gap with other models. And finally, we look at some CRISPR experiments. So here, what is interesting, this is actually a gene level measurement. This is from DepMap, that is based in cancer cell lines, a screen where each gene is knocked down, and you can measure if the cell still lives. And one of the reasons we care about this is because it also suggests ways of, let's say, killing a tumor, like finding which are the genes that you want to, let's say, perturb in order to destroy, to kill a tumor cell. So it's very important. And so many models have been developed to give a gene level constraint score or gene essentiality score, such as PLI, or more recently, GeneBase. And however, we also found that if we use the GPN-MSA scores, it also works really well at this task. So in order to get like a gene level score, we take basically the quantile, a certain quantile of scores along the gene. Again, this is the model. We never thought that it could be used for this, but it seems to be pretty good at capturing functional constraints. So here, I want to illustrate zooming into Nomad, how basically the model is able to score very different types of variants, such as 5' UTR or no-coding RNA. And you can see in many of them, in the majority, it seems to improve quite a bit from previous methods. And I also wanted to highlight like the ability of having a single model for all these kind of variants. It is basically reflected here in the all category, where you can basically contrast all the kind of variants against each other. This is, I think it is realistic. For example, in a rare disease diagnosis, you want to score all the variants along the genome, not just, let's say, UTR variants. And so this ability to contrast all the variants with the same score, I think it is very powerful. So to summarize, on the model development side, this is the first alignment-based DNA language model. And we provide evidence that it improves genome-wide variant effect prediction across clinical, experimental, and population-based databases. So the model is, we found that it's very efficient, like trained in 3 hours. So this is not something that you would care if you just want to use the scores. But if you want to develop, there are so many avenues for improving this model and training it with better data, better like training schemes, better architectures. So I think it is within reach of a academic lab, for example, if you have some GPUs. So some of the open questions, I think maybe the most puzzling for me is that if you include the closest primates, such as chimpanzee, the performance gets worse. And the way I think of it is that the model can just, it is too easy to predict the human if you have the chimpanzee as a context. You can just kind of copy the chimpanzee allele. And therefore, the model doesn't have to learn much during the process. However, we want to use the primates sequences. We know that they are, for example, highly enriching complex disease heritability. So I think this will require maybe thinking more about kind of loss functions that are perhaps phylogenetically aware. And another question is, we only look at this log-odds ratio, but we haven't looked at the embeddings, kind of the latent representation. And we actually found that they help with GWAS variants. So GWAS variants are interesting in the sense that they are under less constraints because they're usually common variants, but still they have a functional impact. And they're using this kind of a similarity in the embedding space helps compared to just using the log likelihood duration. Yeah, so I also wanted to highlight that we have tried to make everything available and including like predicting the score for all possible human like single nucleotide variants, which I think it is pretty cool since there aren't really many like deep learning models that have been able to scale up to all these variants. And yeah, some of the applications that we hope that it can improve are rare disease diagnosis and rare variant burden tests. And with that, I want to thank my co-authors, Carlos, Alan, Cheng Song, and Yun. And yeah, this paper has been recently accepted to a journal, but it's also been on bioRxiv for like more than a year. So feel free to take a look. Thank you very much.
Thank you, Gonzalo, for the really nice talk. Meanwhile, maybe like we see if there are any questions. I can start with one. So I work a lot with like language model on the protein side, and one problem is like with MSA-based model, it's the deep of the alignment. So suppose not in all of cases you can find the same gene or that you can train with the MSA. Is your model dependent a lot on the amount of sequence that you have in an MSA?
That's a great question. So one thing that we tried is, for example, we use the 100 vertebrate alignment, but if you subset it to mammals, I think it does. I think it did significantly worse, but not that much. Maybe it requires more thought. But one thing that I think we certainly find, if you just subsample the variants within the same kind of clades, I don't think it does much of a difference. I think like the main thing is kind of the coverage, how far you can go. And also maybe just wanted to mention, since I'm saying this, like you don't really get, for non-coding regions, you really don't really align more than mammals, usually. There, except some like super conserved enhancers, but usually, yeah, you cannot get too far with alignment. So I think that is one limitation of this approach.
Okay.
Thank you.
And let's see if there is other question. If not, like I have a second one. I wrote another one. So for the test you did, when you compare, for example, to ESM1B and everything, is that a comparison done by using the same dataset? So you rerun ESM1B on the same, like in CleanVar, for example, do you run again like the ESM1B on the dataset that you use, or is like the published results they did?
Yeah, so we took, I mean, they have provided the scores.
Yeah.
And I basically just do like some of a merge of some tables.